Over the past 2 decades, the price of insulin in the United States has increased more than 1,000%. For example, one vial of Humalog insulin, one of the three U.S. insulin companies with monopoly over insulin, used to cost only $21 in 1999. Today, one vial costs $322. For the average type 1 diabetic who relies on 3 vials a month, this totals up to almost $1,000 a month without insurance. With 34.2 million people, almost 10.5% of the United States facing diabetes daily, it is clear that the current insulin market makes such a life-saving medicine unaffordable and inaccessible. The Algae to Insulin project at the University of California, Davis aims to combat insulin inaccessibility by genetically engineering a strain of algae called Chlamydomonas reinhardti to produce a recombinant insulin protein. By taking advantage of this model organism, our project aims to produce recombinant insulin protein while minimizing both production costs and environmental impacts. So why C. reinhardti? C. reinhardti is a eukaryotic, photosynthetic, single-celled organism that requires only a few inputs: sunlight, low filtration grade water, an inexpensive buffer, trace minerals, carbon dioxide, and a nitrogen source. Because of such minimal inputs, C. reinhardti has the potential to become a significantly cheaper biomanufacturing platform. Currently, the most common method of producing human proteins are mammalian cells, such as the Chinese hamster ovary cells, or CHO cells. However, media for CHO cells can often cost upwards to $200 per liter. Cheaper organisms like E. coli can get media prices down to only $4 per liter, but are limited in the type of human proteins they can make. Even Pichia pastoris, a yeast, which has a decent flexibility in terms of protein production, costs $18 per liter to culture, which can quickly add up when manufacturing at an industrial scale. This is where Chlamydomonas reinhardti rises above. It has protein production capabilities approximately equivalent to the yeast of Pichia pastoris, but with such minimal inputs, culturing costs as low as 1 cent per liter of media. In addition, a previous study of protein expression in the C. reinhardti chloroplast has also shown yields of 2% soluble protein, a percentage high enough for commercial protein production. A large amount of potential lies in the protein-producing ability of Chlamydomonas, but how does our team plan to unlock that potential?
Hi, my name is Jasmine, and I will be showing you how the algae to insulin gene genetic construct works. Our project proposal consists of designing a genetic construct, placing it into a free-flowing plasmid, and then into an E. coli. We will then transform our genetic construct that is in a plasmid into the chloroplast of an algae called C. reinhardti. First, we are going to transform and assemble our genetic construct into a plasmid using the slice method. Our genetic construct will arrive into our lab in fragments, and this is to remain cost-effective. 5 to 6 fragments will arrive at a time and will be sliced together. To ensure a high expression of our mini-proinsulin gene, we are using the PSBA gene promoter. This promoter is naturally high in levels of expression. However, our conundrum is that we will have 2 PSBA promoters in the same algae cell, which will then cause our promoter to have low expression. In order to avoid this problem of having low expression, we will delete one of the PSBA promoters along with the PSBA gene and reintroduce it with a different promoter. Since the PSBA gene is necessary for the algae to perform photosynthesis, replacing the PSBA gene's promoter also allows us to perform auxotrophic selection on our algae cells. This is because we can select for the cells that survived without a carbon source. The cells that survived most likely have taken in our inserted genetic construct into their algae's chloroplast, and we can use these for the next step of our project. In addition, our genetic construct also includes necessary parts and components for successful homologous recombination. These include 5' and 3' chloroplast homology regions, a double stop codon, and a polyhistidine tag which will allow for purification in later steps. Once our genetic construct is completed and put together, we can then amplify our plasmid by transforming it into E. coli using a heat shock transformation. This will overall increase our insulin production. Once E. coli has replicated in large quantities, we will lyse the E. coli cells, isolate our plasmid using a mini prep, and then add it into our C. Reinhardti algae cells. We will do this by using the glass bead method to encourage the transformation of our plasmid into the algal cells. Later, we will lyse the algal cells using sonication, and then we will purify our mini proinsulin proteins using immobilized metal affinity chromatography, or IMAC, which is a specialized form of affinity chromatography. To determine if our transformation was successful, we can measure our transcript levels using reverse transcription PCR, which will see if we have the right amounts of genetic construct in our insulin. Following this, we'll use a Lowry protein assay or an ELISA quantification assay to measure the expression levels of the mini proinsulin protein. Using these methods, we can ensure that our genetic construct is inserted into our C. Reinhardti cells.
Hello, this is Joyce Lin, and I'm going to talk about our progress for now and the future plan. The 9 people on the Algae to Insulin team have been working remotely since March 2020 due to the COVID-19 pandemic. Weekly Zoom meetings are held to collect important information and experiment results from related journals. Since we are at the early stage of our project, we are currently working on the protein purification protocol. Moving forward in the future to make necessary preparations to resume working on bench in the fall of 2021, we are going to culture Chlamydomonas reinhardti and insert our insulin genetic construct into the plasmid. The group is understanding the culturing and cloning details right now. According to the CDC National Diabetes Statistics Report 2020, 34.2 million Americans, over 10%, have diabetes, and 88 million American adults, over 34%, have prediabetes. Insulin injection as a supplement for those who have diabetes could control blood glucose levels. By constructing the C. Reinhardti system to produce insulin, we can reduce the cost and waste of producing insulin while increasing the quantity of output. We believe that this research has the potential that can help diabetes patients to live a better life. Special thanks to our instructor, Dr. Mark Fasciotti, and former Algae team lead, Tristan Tran, who helped us to make this presentation. This video was brought to you by Shania Sid De Leon, Jasmine Rettenpaul, and Joyce Pinru Lin. The infographic were from our team members, Maida Ruiz and Anton Block, and the video was edited by me myself, Joyce. All the citations are listed below. We are Algae to Insulin from the Bioinnovation Group. Thank you for your attention.